Welcome everybody to the talk of Charles River, which is about our vision that we have for future QC micro, so quality control micro testing. Before I will speak about our ideas, I would like to introduce to you a little bit Charles River. So Charles River was founded 67 years ago, and we have currently around 7,500 employees with deep scientific capabilities. We are running, meanwhile, 60 facilities in 17 countries. So we are a global company, and we have a unique portfolio for harmonized drug development. We can support our customers with the research models and services at the time of the research, or the very basic research in drug development. Then when it comes to the discovery of new targets, the characterization of new targets, we have the discovery services, and already we can start with consulting and staffing services right from point zero to the preclinical evaluation. Then when it comes to all the clinical trials submission, we have a business unit which is called safety assessment, which is supporting our customers in these activities. And for specific tests, we have another business unit called Biologics Testing Solutions, which is also supporting these activities. The business unit I'm working for is involved in the end of the whole drug development process, which is the manufacturing of the drug and the drug release. Microbial Solutions, that's the name of this business unit, is focusing on three major areas of QC microtesting. So they are also one of the three most important areas of testing. That's microbial identifications and strain typing. If you have contaminations, you should do strain typing in order to find out the origin of your contamination. It's endotoxin testing, so the classical product release testing, and rapid bioburden detection. These three solutions are coming with a package of data management tools. So this gives our customers not only, for example, an identification result, but also the ability to track and trend data whenever needed, whenever wanted. We have a global customer base, and which are making use of these solutions. We are running a lot of studies with those customers in order to prove our performance, but also to find gaps in their processes, which we can use in order to improve their processes with those customers together. We usually do this with one site, but we are trying to find a solution that is applicable to all other sites. So we are thinking about a global solution for the drug manufacturers. Also, Microbial Solutions has a global presence. We have laboratories, so testing facilities in the US, several ones in Europe, but also in India and the rest of Asia. This ensures that a customer who has, for example, sites in Europe, in US and in Asia has the same quality level, because the instruments we are using in these facilities are all the same. The data we are generating is all the same, and the reports also have an equal standard, of course. The Endosafe product line is for endotoxin detection. What we have here in our portfolio is an FDA licensed cartridge technology that allows us to do endotoxin within minutes. We have three different systems. It's called Endosafe PTS. That's a portable testing system that gives you endotoxin testing results within 15 minutes, as I said earlier. If you would like to have a higher throughput model, there's a version called Endosafe MCS. That's it's also a small system that where five PTS testing units are inside the system. We also have a fully automized endotoxin solution, which is called Endosafe Nexus. This is an MCS system connected to a robo. These reagents are of course FDA licensed, and when it comes to product validations, for example, for endotoxin testing, we have the lab in France which is doing CGMP compliant contract testing and is supporting our customers in these product validations. The microbial identification product line is called Accugenix. We are building our proprietary CGMP compliant and validated library since around 15 to 20 years for sequencing, but also MALDI-TOF. We are identifying bacteria and fungi with 16S sequencing or ITS2 sequencing. We are using the ITS2 subunit because this subunit is defining a fungal species. So whenever a fungal species is described, newly described, this is done as a reference by sequencing the ITS subunits, not the D2 subunits. We also offer a service called AccuPro ID. That's a MALDI-TOF identification solution from Bruker, which we combine with our database. And every sample that does not result in a species level identification will be transferred to rDNA sequencing without any additional costs. So this allows us to have a way higher reportable rate than if we would use these systems as standalone systems. I briefly introduced strain typing. Strain typing is a very critical method that is part of investigations, and we are using here single multi-locus sequence typing. This means that we are sequencing species-specific housekeeping genes that are also highly conserved but have variable positions. And this allows us to increase the information we have per sample, to compare the samples with each other and to calculate a genetic distance, which helps our customers in the end to decide whether 2, 3, 4, 5 contaminating samples have the same origin or not. These are the services that are used as an outsourcing solution, but we also have an insourcing solution that's called the Access System. The Access System is a Bruker MALDI-TOF system that is connected to our MALDI-TOF library that is hosted on our server. The advantage of this solution is we are in charge of this ID library. We are in charge of the validation of this ID library, and we are currently doing 1 updates per 3 weeks, where we add taxonomic changes or new species. The customer is benefiting of this, of course, because the species we are adding or the samples we are adding to this library are coming from Access users or AccuPro ID users. So in the end, we are speaking here about a system that is connected to a library which is adapting to the environment of the pharmaceutical industry. This gives us the advantage that we have only relevant species in this library. So if something comes up in the pharmaceutical industry that is unknown, we usually find it out, add it to this library so that an Access user can make use of this entry the next time he would run such a sample. As I said, we have for every product line a data management tool available. In this case, it's a tracking and trending tool, which is part of these solutions. Every identification that is done for our customers, no matter whether they are using the outsourcing option or the insourcing system, is available in this tracking and trending data management tool. You can select here for objectionable organisms and quantify these over time, or if you are interested in how many different species do I have, what's the proportion of these different species within the period of 3 years, 1 month, a week, you can easily do this by selecting the right parameters and filter for that. The benefit of this tracking and trending management tool is it's facilitating heavily the tracking and trending duties your QA has on the one hand. On the other hand, you can export all those reports and integrate them into your LIMS system. We recently acquired a company called Celsis. So Celsis helps us to have also a rapid micro detection solution. We have two systems here. One is the Celsis Advanced 2, that's the larger one. It has a high throughput with 120 assays per hour. Both the mid-range throughput with 30 assays per hour is the smaller system called Celsis XL. And these systems are using bioluminescence. So a luciferase assay. The advantage of this assay is it's fine-tuned. So there is a reagent which is reducing the environmental ATP, which means we are reducing the background. And in parallel, we are amplifying the actual signal that comes from the luciferase. So we have a better signal to noise ratio, which is increasing the specificity of these assays. These assays are qualitative, so you only get a yes or no answer. And this is why we are currently working on, this is still in the beta stage, on a system called PTS Micro. That's that one here. The PTS Micro system is using a different assay, gives you a result in 30 minutes, and is a quantitative system. So this system automatically counts the cells that are in your product. We know that rapid microdetection systems require quite an, sometimes extensive validation, because one needs to show that these systems are equal or better than the compendial methods. And also here we are offering support on the validation end, but also when it comes to regulatory questions. So in the end, Microbial Solutions is dealing with 3 routinely separated tests, but these tests have one common scope, and the common scope is the environmental control and final product release. When we go to our customers and ask, how is your QC microtesting connected to the rest of your facility, we see that picture. So we have the manufacturing, we have a central QC lab, which is totally separated and mostly divided in a bioburden testing group, an endotoxin testing group, and a microbial identification testing group. Moreover, each group is using different suppliers. So this does not really ensure that you have a harmonized quality level already within one site. So how should you have it then on a global level? On the other hand, manufacturing is providing the samples to be tested for the product release, of course, at the very end of the production line. And then the products, for example, for bioburden testing are getting incubated for 3, 5, or 7 days, depends. And this is a waste of time in the end, because the product is actually finished, it would be ready to be released, but you need to keep it in your warehouse. This is costing money. On the other hand, if there are reasons for process improvement that you would like to work on with R&D process development, or if you have further downstream activities, although these decisions are pending on the decisions coming from the central QC lab. So we have here a clear separation of the whole process, and this does not offer a continuous analysis and control of manufacturing processes based on real-time measurements or rapid measurements while the process is ongoing. The limits of this approach is that you have increased manufacturing lead times because the test methods are not linked with each other. You have a reduced environmental control because you wait all the time for the results while the process is running, and therefore an increased or potentially increased contamination risk. This is, on the other hand, also reducing the lab efficiency. And when we speak about an harmonized quality level, several suppliers are impeding this goal. When we take these 4 points together and are looking at the financial side of things, we're speaking here about increased costs due to inefficient micro QC testing. So we were asking, how can we overcome these obstacles? And for us, the solution is PAT. PAT is process analytical tools. This is a framework which is based on innovative pharmaceutical manufacturing and quality assurance, which is based on scientific principles and tools that are supporting the innovation, supporting your process, and have the goal to improve the process while it's running. The definition of PAT is these are systems for continuous analysis and control of manufacturing processes based on real-time measurements or rapid measurements during processing of quality and performance attributes of raw and in-process materials, and the important point, processes to assure end product quality at the completion of the whole process. So transferring the PATs into a QC microtesting means that one needs to have instruments, tools, processes available that allow for an online, inline, on-stream, in-stream, real-time or near real-time solution for monitoring continuous or episodic operations. What happens in the end that an appropriation implementation of PAT requires a transition from a summary batch information to a stratified in-process batch sampling and testing. Those PAT tools require, of course, a detailed understanding of your processes and of your control mechanisms. Therefore, modern process analytic tools are required, and because we would like to monitor in-process and the end product, while we are also have the ability to continuously improve the process itself. Charles River has those PAT tools for QC microtesting in its portfolio. We have on the CRL testing side the rapid endotoxin testing systems where you get a result within minutes, so you can easily use it while the process is running. We have for the microbial detection two different solutions that are both non-destructive and qualitative or quantitative tests. Non-destructive is important here because if you realize during your process I have a high bioburden load, or I see something which I see some contamination that should not be there, you can culture these contaminations and hand it over to the microbial identification. And also with a MALDI-TOF, which is backed up or linked to our library, I said the library is continuously improving and it's validated. You can have your ID results also within minutes. So also you will have an ID result right at the beginning of the investigation. Why should one use our PAT tools? First of all, the information or in-process control information and end product information is very critical. And these systems give you a fast, decisive, qualitative or quantitative results that allow you a rapid release or a rapid response to any process deviations. The systems have a broad range of applications. They are validated to hundreds of products. So if a customer comes to us and asks, could you please help us with a validation of your systems to a product? Yes, we can. This is further increasing the lab efficiency. We have minimal requirements for these systems. They are very small, if not portable. They are really easy to learn and easy to use. They are reducing the number of tests you have to do and thereby are reducing the waste. We also have, of course, the technical expertise behind. We have a strong technical support that is able to help you with any of your testing results for those 3 product lines, and also to help you to make more out of your data than only having an identification report or a certain concentration of endotoxin. We would like to unify these 3 testings into one solution, and the key here is, of course, the data management. We would like to come up or are working on a solution where we are unifying endotoxin testing, micro-ID and bioburden testing results within one database. And please keep in mind, these systems are very fast. So the database is continuously fed with data while the process is running. The database is or should be accessible by the QA or QC reviewer, or even by a corporate office who needs to make fast decisions on a global level. Just come back here to the conventional QC microtesting model. I said we are separating here every process and we are waiting for results. And this is exactly the opposite of what I just presented to you. So if we are able to implement this future data management to unify these three testings, we come up with a lab of tomorrow via process analytic tools. The core of this lab of tomorrow is the QA/QC review. We have the same systems in all 4 testing units. That's manufacturing, central QC lab, R&D process development, possibly, and downstream processing. And the samples are tested at the point of collection, and the results are sent to QC and QA for review. As all units are using the same systems, we have the same quality level. And more important, we have real-time results, so we have real-time data flow here from all 4 units into QA/QC. In the end, we can come up with a continuous analysis and control of manufacturing processes based on real-time measurements or rapid measurements during processes. We would have all the data we would need to have available for product release at the end, right at the end of the production. So this is reducing the warehouse space and finally the production cost. So the Charles River Lab of tomorrow is reducing or will reduce the risk due to real-time accurate results. It will facilitate and accelerate the decision-making processes. It will proactively improve the manufacturing environment because it gives you the ability to, based on the data that is coming real-time to the QA/QC, to adjust your process if needed. It will help you to reduce your manufacturing lead time, and on the other hand, leverage and harmonize the quality level, not only per site, but also on a global scale. In the end, again, you can reduce your production costs. So, thank you for your attention. If you have questions, you can visit us at our booth at 3G 105, and we can talk about these systems if you like, or about the whole idea that we have.